Yes, this metabolism, how to write. So here we are going to see how to write the chylomicron metabolism. I am going to give some titles or tips for the chylomicron metabolism. First one is formation and composition of nascent chylomicron from intestine, right? Second one is formation of chylomicron, that is nascent means immature chylomicron. Chylomicron means complete chylomicron, matured chylomicron, from nascent chylomicron and HDL, because HDL releasing 2 apolipoproteins for the formation of chylomicron. Third one is, what is the role of LPL, that is lipoprotein lipase, very important enzyme for the activation of triacyl glycerols converted into fatty acids and glycerol, right? The glycerol is for the liver for the glycolysis or gluconeogenesis. Free fatty acids deposited in the peripherals like adipose tissue and gives energy to the muscles. Next, releasing chylomicron remnant with the uses of TAG and cholesterol. The next one is degradation. After the remnant chylomicron formation, how it will enters into the liver for the degradation in liver through receptor mediated endocytosis. Next one is site of chylomicron metabolism occurs in adipose tissue and skeletal muscle. Role of heparin, this is very important viva question or sometimes MCQ question, that is lipolytic activity of heparin. For myocardial infarction patients, they are giving heparin drug for the treatment, which is having lipolytic activity in myocardial infarction. The lipoprotein lipase is stimulated by insulin. This is also another important MCQ question. Insulin has a role of stimulating LPL, lipoprotein lipase, and increases the lipolytic activity for the myocardial infarction patients. The last one is, if you are having in the laboratory lipemic sample, means that is upper portion of the container or in the sample white milky color or lipemic sample, means the patient is having more amount of, that is 600 milligram, 400 milligram, 800 milligram of triglycerides in their blood, right? Hi students, today we are going to discuss about one more important 5-mark short notes from lipid metabolism. The topic is chylomicron metabolism, right? In lipoprotein metabolism, we have chylomicron, HDL metabolism, LDL metabolism. All the three are your important 5-mark short notes. Before going to this chylomicron metabolism, you should know two important points. That is, one is composition of chylomicron. Second one is apolipoprotein involvements in chylomicron. These two factors are very important for your chylomicron metabolism, right? The first one is, from the diet you will get cholesterol and fatty acid content like lipids enters into the small intestine. So chylomicron metabolism starts from intestine only, right? So through lymphatics they will go to blood circulation like portal vein, you know the lymphatic circulation. So first formed compound is nascent chylomicron, that is incomplete chylomicron or immature chylomicron, right? Here the composition is triacyl glycerols, cholesterol and phospholipids, whereas apolipoprotein is only 2A1 and B48, okay? So from this nascent chylomicron, which will be converted into complete chylomicron or matured chylomicron, okay? Here you can see how many apolipoproteins are there: B48, A1, C2 and E. That is why this C2 and E is derived from HDL. That is why with the help of HDL only the chylomicron formed from chylomicron remnant. See, all of you listen, don't worry about to see the diagram, get scared about the diagram. It is very easy only. First one is intestine from diet, second one is nascent chylomicron, third one is chylomicron formation. Don't worry, right? So here composition of chylomicron is the same: triacyl glycerol, cholesterol and phospholipids, whereas the apolipoprotein is increased to 4, that is A1, B48, E and C2, right? After this chylomicron formation, these triacyl glycerols are converted into free fatty acids and glycerol by important enzyme is present in the vessel wall of the muscle adipocyte tissues and skeletal muscle, cardiac muscle, like that, lipoprotein lipase. What this lipoprotein lipase will do is, they will be liberating free fatty acids to the adipocytes and muscles and glycerol to the liver. They have two separate important functions. This free fatty acids gives energy for the muscles, whereas this glycerol enters into the liver for the purpose of, depending upon the concentration, they will be go to either glycolysis or gluconeogenesis. If there is no glucose, means this glycerol is used up for glucose formation by gluconeogenesis. If there is a need of ATP or energy, means they will be entering into glycolysis after the conversion of dihydroxyacetane phosphate and then enters into glycolytic pathway, right? Like that, this triacyl glycerol, major amount 90% of chylomicron composition is TAG. This TAG is converted into free fatty acids into muscles and glycerol into liver. This point is very, very important. And this lipoprotein lipase is absent in liver. This is very, very important MCQ, right? After this releasing all this, they will be named as chylomicron remnant, the remaining portions like cholesterol esters, cholesterol phosphates, triacyl glycerols in lesser amount. Okay, because most of the triacyl glycerols are converted into fatty acids and glycerol by LPL. Here the cholesterol composition is very less. All the composition is very, very less compared with the chylomicron. Here the apoenzyme is only 2B48 and E. This E is very, very important for the receptor attachment in the liver. Before that, from the chylomicron, again the ApoA1C2 cholesterol again enters into HDL for the recycling. That is why cholesterol content is low, and TAG is almost 90% released for these peripherals and 10% only available in the chylomicron remnant. After this chylomicron remnant, they will be entering into liver for the degradation. Here the degradation name is receptor mediated endocytosis. You all know the endocytosis in cell membrane and transport systems by the lysosomes. So we have two mechanisms for the degradation of the chylomicron remnants. One is hepatic lipase, another one is remnant receptors, which is available in the liver. So after all these triglyceride, free fatty acid, cholesterol, phospholipid, cholesterol esters in the liver, by the degradation by the receptor mediated endocytosis, after that the receptors are ready for the next cycle, right? So don't worry about chylomicron metabolism, very, very important metabolism, because the function for this is transport the triacyl glycerol from intestine to the peripheral tissues. That is very, very important answer for your viva. What is the role of TAG? Transportation, what is the role of chylomicron? Transportation of triacyl glycerol from intestine to the peripheral tissues, right? Is there any clinical involvement? Yes, we have one disease, A-beta-lipoproteinemia in the chylomicron metabolism. This is not significant, but we should know about the name of the disease, A-beta-lipoproteinemia. Okay, bye.